medicine in the physiology department, and I'm also the director of the honors medical research program. So today we have the final presentations for all these fine students who participated, or at least the second half of them. We had half of them yesterday. And so I also want to acknowledge Ms. Latora Rogers, who is a Health Sciences Academy counselor, and without whose help this program would not get off the ground, because she is way more organized than I am. So thank you to you. And without any further ado, first up today we have Alexia Vachhani and Paulina Weglarczyk. And I apologize if I mispronounced any names.
Good afternoon. My name is Alexia Vachhani, and I am from D.H. Conley High School.
And I'm Paulina Veglarczyk, and I go to Rose High School.
Today, our presentation is the pH sensor GPR68 inhibits vascular smooth muscle cell growth through cytoskeletal vasodilator stimulated phosphoprotein.
In our lab, we studied cardiovascular disease. And cardiovascular disease is the leading cause of death in North Carolina and all around the world. It makes up over 30% of all global deaths. So the cardiovascular system contains the heart and the blood vessels, and the heart pumps blood throughout the blood vessels, and it carries blood, and blood vessels carry blood throughout the rest of the body. Some terms that we went over in the lab are ischemia, hypoxia, and acidosis. So ischemia refers to a reduction of blood flow flowing through the blood vessels. And you can see on the picture to the left, there's a blood vessel and there's an occlusion or a blockage in the blood vessel, causing a reduction of blood flow getting to the downstream tissues that blood vessel leads to. Hypoxia refers to a reduction of oxygen. So with the reduction of blood flow comes reduction of oxygen. And acidosis relates to both of these terms because with the reduced oxygen getting to the tissues, there's a buildup of hydrogen ions in the extracellular matrix of the cell environment, and that causes an acidic environment and a higher pH in those tissues, which is acidosis. And in our lab, we studied GPR68, which is pH sensing or acid sensing, and it responds to these acidic conditions.
So GPCRs. What are GPCRs? G protein coupled receptors. These are 7 transmembrane receptors which react and are very critical for cell signaling. Within GPCRs, there's a receptor called GPR68, which we study in the lab. It is, as Paulina mentioned, sensitive to acidosis. So we study GPR68 and its downstream signals. So this is a diagram that we produced in our lab. As Paulina mentioned, you can see the hydrogen ions in the top of the diagram in the extracellular matrix, and an increase of these hydrogen ions causes acidosis. The blue in the diagram is the cellular membrane, and then the black is the receptor itself. So once these alpha, beta, and gamma G proteins are activated, it goes through a downstream signal through cyclic AMP and then pKa to then reach VASP, vasodilator stimulated phosphoprotein, which is the protein we study in our lab. So there are a few main questions that we were researching. The first one was the impact of the pH sensing GPR68 on proliferation, which is unknown, and then also vasodilator stimulated phosphoprotein, or VASP, which is the downstream signal through pKa, but we don't know the correlation between GPR68 and VASP. So we don't know if VASP is the one causing the decrease in cell growth through GPR68.
We had 2 hypotheses in our experiment. So the first was that GPR68 will inhibit growth in vascular smooth muscle cells in acidic conditions. Our lab had preliminary data that shows GPR68 is increased in acidic conditions, but there's no research showing that GPR68 directly controls vascular smooth muscle cell growth. And our second hypothesis was that GPR68 will inhibit growth of vascular smooth muscle cells through VASP. And this was used to see if VASP is the direct mechanism that GPR68 works through to inhibit growth, because that's not shown in any research either.
So combining our hypotheses, one hypothesis, which is GPR68 has the capacity to induce VASP expression and inhibit cell growth during the vascular smooth muscle growth process as a part of the disease process. So we had two main aims. Aim one was what Paulina was studying, and that was to determine the role of GPR68 in cell proliferation. And then aim two, which is what I was studying, was to analyze the expression of VASP as a downstream signal of GPR68. For both of our experiments, we used rat vascular smooth muscle cells from the tunica media of the thoracic aorta, and both of our experiments were conducted and analyzed in normal and acidic pH media.
Alexia and I both did a lot of cell culture in the lab, and I did a proliferation assay like she mentioned. So we used primary rat cells that the lab had already generated. They were in a freezer, and we grew them in flasks, which you can see on the left. That's a flask with cells and media inside. The media is the red solution, and that provides a good environment and nutrients to the cells so they can divide and keep growing. And once the cells were confluent enough in that flask, which means they took up a lot of the surface area and were growing, we passaged them to a 12-well plate, which you can see on the right. That's divided into normal and acidic media. But we let the cells grow in the wells in that 12-well plate, and then we quiesced the cells, which means we added quiescent media, and that made sure that we're ready to start the experiment because the cells would be growing at the same rate and they would be at the same level in the cell cycle. And once we did that, we let the cells sit in the quiescent media, and afterwards we added either acidic or normal media. So in the other slide on the 12-well plate, you can see that it was divided into 6 wells of acidic media and 6 wells of normal media, and they were divided like that in one well just for consistency. But we had 2 of those plates, and one was used for the 48-hour time point and one was used for the 72-hour time point. And our pH of the normal media was 7.4, which resembles the pH of the human body in a normal condition. And then the acidic media was at a pH of 6.65 to 6.72. And we grew the cells in the acidic and normal media for the 48 and the 72-hour time points. And afterwards, we took a sample out of each well, and we ran that through the VASP machine, which you can see on the right. And VASP machine gave us our total cell number count and our viable cells, which we used to make our results. So the 2 graphs on the left of the slide are preliminary data that was generated in the lab before we got to the lab, but all of the graphs are correlated in a way, so I'll go through each of them individually. The first one on the very left shows GPR68 gene expression in wild-type normal and wild-type acidic mouse cells. And you can see that the wild-type acidic mouse cells have a much higher GPR68 expression, so this links GPR68 expression to acidosis and shows that GPR68 is increased and activated in acidic conditions. The graph in the middle shows wild-type and knockout mouse cells. So wild-type mouse cells are just normal mouse cells, but the knockout mouse cells don't have the GPR68 gene because it's been knocked out. So we expected to see the knockout cells have much more growth because GPR68 isn't there to inhibit growth. And that's what this graph shows. It shows that GPR68 inhibits vascular smooth muscle cell growth. And then the graph on the right is what we generated with our data. And it shows the normal media on top and the acidic media on the bottom and cell number on the y-axis. And so you can see that in acidic media there's a reduced amount of growth, while in the normal media there was much more. So this shows GPR68 is activated in acidic conditions and inhibits growth.
So another procedure that we used in the lab was Western blotting, which is used to find the proteins in a specific sample, but using their molecular weights. But before we actually conduct the Western blot, there are 2 main procedures that we have to conduct. The first one, so as Paulina mentioned, we grow our cells in flasks and plates. So we conduct a series of procedures to remove those cells from those plates and wells and transfer them into Eppendorf tubes. And so that process is called homogenizing. And then we conduct a Bradford assay, which is, so we take the samples inside the tubes and we check each one to see how much protein there is in each one. And so we get protein quantities, and then we're able to analyze those to find out how much of each sample we use to get the same protein level. And then we're able to conduct the actual Western blot. So we take the quantities that we previously produced and we inject them into a gel, and then we run it through a electric gel shock. And so that is the gel electrophoresis portion of it. And then we analyze the images, and then we are able to probe for the specific protein that we're looking for. And to the right, you can see the instrument that we use, the ChemiDoc. On the left, that is how we lay the gel inside of while conducting the Western blot. So this is my total protein Western blot before probing for VASP. So as you can see, we did 3 lanes of normal and then 3 lanes of acidic and then 3 normal and 3 acidic, and that is to maintain consistency throughout the whole gel. So you can see the darker lanes were the normal cells, while the acidic, you can't really see the actual proteins. And so there are 3 main specific molecular weights that we're looking for: 75, kilodaltons, 50, and 37. These are images from someone previous to me in our lab, and they were also probing for VASP. So the top 2 images, you can see that those are the VASP expression, and the bottom are total protein. So this is comparable to the image that I showed you in the previous slide, and then the top is what they probed for VASP. This experiment is a little bit different because they were looking for the difference in the passage. So when you move cells from one flask to another or change their environments, it's called you change a passage level. So they were just checking that. But you can see that the total protein, these images, the bottom images, are comparable to the ones that I showed you previously.
So with the proliferation assay and Western blots that Alexia conducted, we were able to say that our data suggests that GPR68 does inhibit growth in vascular smooth muscle cells in acidic conditions, which goes with our hypothesis. So we saw that cell numbers decreased in acidic media because GPR68 was activated. But one thing we couldn't find or figure out was the exact impact of VASP on GPR68 because we had some technical difficulties with our Western blotting. But in the future, we hope to use this data to create therapeutic treatments that activate GPR68 or downstream signals like VASP, and therefore treat abnormal cell growth and cardiovascular diseases.
So throughout our time here, we had a few challenges and limitations, but we were able to solve the majority of them. So the first one, originally when we started working in the lab, we had a difficult time growing our mouse cells, but we switched over to rat cells and those grew fine. And so all of the results that you've seen throughout the presentation have been our rat cell data. And another thing is that, as Paulina mentioned, we had a few technical difficulties with our Western blotting. So with our VASP hypothesis, the data is incomplete, but we do have the total protein blot that I showed you earlier. And also, the pH of our acidic media was increasing over time, but we also titrated acidic media into there to keep steady pH level, and so we were able to solve that as well.
So for our future goals, I will be attending East Carolina University in the fall, and I'll be majoring in public health with a concentration in pre-health professions. I will be also going to East Carolina University, and I will be double majoring in computer science and biology. And I had a really great time in the cardiovascular section, so that may be my future direction. Yes, we both had a great experience in the lab and hope to do lab research in the future.
This is just a few of our references that we used throughout our presentation, as well as our time here during our research. And lastly, we'd like to make a few acknowledgements. First, Ms. Rogers and Health Science Academy for arranging this whole program. Dr. Walters from Brody School of Medicine for organizing everything, the whole program, and arranging for all of our seminars. And last but not least, Dr. Tulis and Madison, our PhD student and preceptor, for everything that they've taught us. We're so thankful for everything that you've done, and we'll take these experiences forever, a lifetime with us. So thank you for your time. Do you have any questions or comments?
You said the rat cells do better, maybe it's for why?
Well, I think the rat cells that we used were on the 10th passage already, so I mean they were generated before we got into the lab. And I think the mouse cells that we were using were just brought after surgery, so they were pretty new, and so maybe that could be the reason why, but I'm not exactly sure about that.
Any other questions?
So since there is a difference between the mouse and the rat cells, do you think there could be a difference between the rat and the human cell?
Yes, yeah. But mouse cells are usually used for like human, or compared to human
Thank you.
Thank you.